Let's talk about everything that you need to know about cell division. So basically, let's take this rugby player, okay, and we zoom into his body. We look at what he's made of, and we know that it is cells, right? He's made up of billions of cells. Now, the thing is, cells have a lifetime and they will die after a certain period of time. So if our cells just kept dying and dying, we would pretty much turn to dust, right? So it must be that our cells can replicate or divide or make new cells. Otherwise, we wouldn't be able to maintain our shape, our size, right? So that's true. So cells do something called cell division that maintains our bodies. They keep replicating and replicating so that we never just fall apart, okay? Now, what is the purpose of cell division? Why do our cells need to divide? For one important reason, simply growth. How do we turn from a baby to an adult? How do you turn from a baby to this massive muscular guy? You gotta make more and more cells. You gotta become bigger and bigger, right? So that's one purpose of cell division. Now, that's why when we get older, we kind of get a bit shorter, we lose hair because our cells don't divide as much anymore. Our body gets old and the ability for our cells to divide decline. Another important scenario is pretty relevant to rugby players because this guy right here is a rugby player, right? And we know that as a rugby player, it's a contact sport. You get injured really quickly, a lot of bruises. Now, bruise is pretty much your skin cells being wiped off, being rubbed off. So cell division is important in healing to replace those cells that have just been scraped off, right? So that's another purpose of cell division. Now, what does it mean for our cells to divide? When I say our cells divide, what do I mean? Like, how does it look like? So, it means that our cells split in half like this. No, I'm joking. It's not what I mean. This is not cell division. This cell division is not when our cells just split apart like that. It's definitely not like this. Like one cell doesn't get half a nucleus, the other cell doesn't get half the nucleus. Like this doesn't happen. No. How about this? No, this is also not true. Like one cell getting almost nothing, the other cell getting almost everything. No, this is not cell division. So how about this last one? Yes, this looks right. So cell division is when our cell goes from 1 to being 2, okay? It basically divides from one cell to being two genetically identical. This is very important. They have the exact same DNA, okay? They're the exact same cell, okay? Now, that's what I mean by cell division, turning one cell into two cells, okay? And then these cells can divide once more and become two cells themselves, okay? This is cell division. Everything, all the organelles, all the DNA, everything remains exactly the same as the parent cell or the original cell. Now, basically, a cell has a cell cycle. So you know how humans, right? We have a life cycle, right? We start off as babies, we grow up, get a bit older, develop some features, maybe we grow some facial hair, stuff like this happens, and then we get old and we die, right? Now, cells have a similar cycle. They also have a cycle, and we call it the cell cycle. Basically, it starts off with something called, let's go simple, okay? The cell cycle consists of two big phases. Interphase. Okay, this is the longest phase. You can see it's long. And then there's cell division. Okay, so it's got two phases. Interphase is like the living phase. It's the phase where the cell is kind of, when you look at it, it looks the same. It's just living, okay? It's the living phase. And then there's the phase where it divides to become a new one, okay? That's called cell division phase. So let's describe a little bit more, okay? Because you need to know this. Interphase is divided into three phases: G1 phase, S phase, and G2 phase. Okay, I'm going to describe exactly what happens here. So let's take a cell. Okay, the same cell we've been looking at. Okay, G1 phase stands for growth phase. G for growth. Let me show you. Growth. Okay, we're going to write it right here. Growth phase. Easy to remember. And three things happen to the cell here. Three important things. Okay, just gonna zoom it in. One is growth. Okay, it's gonna get bigger. Okay, it's gonna grow. It's gonna turn from a smaller kind of cell to a bigger kind of cell. Just simple growth. All the organelles get bigger, the cell gets bigger, and all this. Secondly, it makes cell replication proteins. So basically, it's going to make this cell is going to make some proteins. Let me show you. It's going to make some proteins that allows it to be able to divide later. So remember I said there's interphase and cell division phase. Now in this phase, it does growth and it makes cell replication proteins. These proteins are used later in mitosis, okay? Because in the beginning, right after, like in the beginning, there's none of those proteins, so the cell can't divide yet. So this phase is important to make those proteins so that later on, mitosis or cell division can happen efficiently. And the last thing that happens is it makes more organelles. Why does it do that? So that when the cell divides later, the two baby cells both have enough organelles to survive. So let me show you. So here we have the original cell. You can see it grows and makes a lot of proteins, and also the organelles increase. So here we only have 3 mitochondria, and then here we have many, many more. So the organelles get more, okay? Even all the other organelles, I just didn't put it here, okay? Because now when the cell divides a bit later, both cells will have 3 mitochondria, okay? Just like the original cell. So that's it. That's all that happens during G1 phase. How about S phase? So S phase stands for synthesis. S for synthesis phase. So what is being synthesized exactly? DNA. DNA is being synthesized because remember, our original cell, right? Our original cell has DNA, right? And we know as a human, because we're talking about a human here, right? We're talking about this guy. As a human, he has 46 chromosomes or 23 pairs, right? And each pair has 1 chromosome from the mom, 1 chromosome from the dad, right? And we know that when this cell wants to divide, if it divides with that DNA, then the daughter cells or the baby cells will get way less DNA than the original cell, right? Because if this cell has 46 chromosomes and it divides in 2, now the daughter cells will have half of that. So we got to make sure that the daughter cells will have the same amount of DNA as the original parent cell. So we got to make sure to double the DNA because we doubled all the organelles, right? But we also got to make sure to double the DNA. So that's what happens during this S phase, the synthesis phase. We times 2'd, we doubled the DNA because later when the cell is going to split during mitosis, the 2 daughter cells will have the right amount of DNA, okay? Now, let me give some more details. In here, like I said, we have 23 pairs of chromosomes, okay? In total 46. And each pair has 1 from the mom, 1 chromosome from the mom, 1 chromosome from the dad. So we're taking here, we took out of the nucleus 1 pair of chromosomes, okay? 1 from the dad, 1 from the mom, And what I mean by when we duplicate it is if the DNA splits right now, the 1 cell will get the dads and the 1 cell will get the moms. And that's bad because we need the baby cell to have both the mom and the dads, just like the original cell. So when I say we duplicate it, I mean we're going to double this. So we're going to get 2 of these now and 2 of these, so that when the cell splits later, the daughter cells can each get 1 of these, okay? And that way it will be the same as the original cell. Okay, real quick here. Let's label some keywords. Now, what do we call the space in the center? The space where these 2 chromatids connect, this space. We call that the centromere, okay? That's going to be important for later. And because these 2 chromosome pairs, have a similar function, because this one's from the mom, this one's from the dad, they still do the same thing. For example, this chromosome might be its function may be to regulate hair color, okay? And this is the one from your mom, and that's the one from your dad. So we call them homologous chromosomes because homo means the same. They have the same purpose. We call them homologous chromosomes. They have maybe they have slightly different features because one is from your mom and one is from your dad, but they still have the same general purpose. We call them homologous chromosomes. Now, when we duplicate it, so we just duplicated it, right? Let me move this a bit up. We now duplicated the moms and the dads so that when it divides later, the daughter cells or the baby cells will have the right amount of DNA. Now, what do we call it when this one duplicated itself? Now we call these 2 sister chromatids because they are sisters. They have the exact same DNA. They're the exact same thing, just duplicated. So we call them sister chromatids. Okay, so those are just some key words that you need to know. Now that we're done with the S phase, that's where DNA is replicated, let's go into G2 phase. So G2 phase is very, very, very similar to G1 phase. There's some more growth and some more organelles being synthesized and some more proteins being made to prepare for mitosis. So it's essentially the same thing. Well, we lost an arrow. Anyways, let me just copy an arrow. So now this one will basically duplicate. There'll be more organelles and the cell will be even bigger. Now it's ready for mitosis. So we just covered this whole long part. You can see mitosis is actually quite short. It's a short part of the cell cycle. This whole interphase is the long phase. The whole phase preparing and growing for mitosis. That's the long part. Mitosis is quite short. Now, mitosis. We're getting now to the cell division part. Mitosis can be divided into 4 stages. Prophase. First of all, before I do that, what is the purpose of mitosis? Mitosis is the stage that is responsible for dividing the nucleus, okay? Because we just saw, we doubled the DNA, we doubled the nucleus, the amount of stuff in the nucleus, the DNA. Now, mitosis is the part that's going to divide this nucleus into 2 separate compartments, okay? It's going to divide the nucleus into 2 separate compartments. And then cytokinesis is the process happening right after mitosis that is going to divide the cell into 2, okay? So mitosis divides the DNA into 2 compartments, so now we have 2 nucleuses, and then cytokinesis is going to divide the entire cell into 2 so that we have 2 separate cells, okay? And that's the final part. Now, like I said, mitosis has 4 phases: prophase, metaphase, anaphase, telophase. Let's get into this right now. So to make this simpler, okay, we've been talking about this cell. I've made a little cell that is far simpler because I'm ignoring all of the organelles because they don't matter now, okay? We're just caring about the DNA. That's the key part. So you can see here on our cell that we talked about so far, this was the cytoplasm. In this new cell I'm going to use, this pink part is the cytoplasm. And this inside part here is the nucleus, okay? I made the nucleus quite big because there's going to be a lot going on in here. So just notice what I'm doing, okay? Don't be confused when you see this cell. It's just a very simplified version, ignoring all the organelles since they don't really matter for now, okay? So let's get into it. So we start off, okay, right here. We start off with this cell, okay? The nucleus and the cytoplasm, right? Now, we cannot see the chromosomes, okay? We cannot see it yet. It is non-condensed, meaning it's not condensed, okay? So if it's not condensed, we can't see it. Now, why is it not condensed? It is not condensed because it's being used, okay? When DNA is not condensed, so it's like a super thin string, that means it's being used for transcription and translation to make proteins, okay? Because when it's condensed, the molecules in the cell cannot use it, okay? It's hard to use. So during the non-condensed phase, when we cannot see the chromosomes, it's being used to make the cell survive and do a bunch of stuff, okay? Now, we are now going to enter prophase, okay? So what I was talking about just now was when it's right before prophase. So now we get into prophase. And prophase, 3 important things happens, okay? Prophase, we're going to remember because it's the first part. Pro means before everything else, so it's the prophase, okay? And what happens here is 3 things. The DNA will condense, so these chromosomes will start being visible. Remember, just now in this interphase, we doubled the DNA, right? So we had 1 from the mom, 1 from the dad. We doubled it so that now we had 2 from the mom and 2 from the dad. So when the cell divides, the daughter cell can get 1 of each. So notice these 2, okay? I'm going to use these because remember, in reality, we have 23 pairs. I'm going to demonstrate this process of mitosis just with 1 of these pairs because it's the exact same for all of the other pairs. So if you understand it with 1 pair like this, you'll understand it for the rest. So you can see here, I have these pairs. We duplicated it, now we have 2 from the dad, 2 from the mom, okay? But it's the same for all of the other 22 pairs of chromosomes. Now, so that's what happened. The DNA is supercoiled or condensed because now it's tight and easy to see and easy for the cell to move them into 2 separate compartments, okay? That's why. And you notice the membrane, the nuclear membrane starts breaking apart a little bit, okay? You're going to see why later, but it starts breaking apart a little bit because here it was still fully formed. So we notice we have double the DNA, as I mentioned before, from the interface. And now the centrioles, these little star-like things, start to form. And you'll see why they are important. They start to form and they start moving around. And now we enter the next phase called metaphase. M for middle. M for middle. In this phase right here, in reality, the chromosomes can be anywhere, okay? They're not really organized yet, but in metaphase, they will line up in the middle, just like this, okay? Because you'll see why. And these centrioles will start moving to the opposite side. So one will go here and one will go there. And then you see these little fibers, these little strings will start forming. They are called spindle fibers. Spindle fibers, okay? These spindle fibers are gonna grow and grow and grow and grow and attach right here. What do we call this spot again? Let's go back. We call it the centromere because it wants to grab right here so it can pull apart these two sister chromatids, okay? So that each of the baby cells can get one, okay? So yeah, it keeps going, it grabs onto the centromere, and that's what happens during, and you can see now the nuclear envelope is completely destroyed. It's not there anymore. And this is done so that these chromosomes can easily move around and go to where they want in the cell. There's no restriction, there's no barrier, okay? It makes it easy for all of your 23 chromosomes to line up in one line, right? Because we know we have 23 pairs. I'm just using one important one here, but we have so many that's gonna line up in the middle. And if this nuclear membrane is restricting them, then they can't line up properly. Okay, so that's metaphase. Remember, M for middle, they line up in the middle. Now what's next? Next, we get to anaphase. So anaphase, you can think of as a girl, Anna, okay? Her name is Anna. And during this phase, you have a girlfriend, right? Or you're gonna just think about it, Anna is a girl. And you're a guy, okay? Pretend you're a guy, even if you're a girl for now. And you're dating this girl, and now it's time. You've known each other for too long, and you didn't really like her, and you gotta break up now, okay? So this is called anaphase, the phase of breaking up with Anna. So these spindle fibers start shortening, they start contracting and pulling back on these sister chromatids until they break apart. Notice now, we have one from the dad, one from the mom, going to one side, and one from the dad and one from the mom going towards the other side, okay? This is anaphase. Okay, and that continues to happen. And now we get to telophase. And you can see, what's the difference? You can see the nuclear membrane starts forming. We're starting to form another nucleus, okay? Two separate nucleuses. One is being formed here on one side of the cell, and one is being formed here on the other side, these two nucleuses. And they both have the same DNA, the exact same DNA. And why is telophase easy to remember? Because it looks like a telephone, right? An old-fashioned telephone. One nucleus here, and then the phone, and then the other nucleus here, okay? Easy to remember. This is the end. Division of the nucleus. The nucleus is now divided into two. So notice, prophase, metaphase, anaphase, telophase. We just covered all that you need to know about mitosis, okay? Because the next phase is cytokinesis. This is the process where the cell actually divides into two, because now we only divided the nucleus so far. So this brings us here. Now we have cytokinesis. This is after mitosis. And this, now you can see here, the membrane, the cell starts to divide into two. The cytoplasm starts to divide into two. And this brings us two separate daughter cells, or two separate baby cells. And again, they have the exact same DNA as the original cell, okay, as the original cell. Okay, because remember, our original cell here only had these two. We had to duplicate it so that when it splits, the baby cells have the exact same as the original cell. Okay, so what's going to happen now is this cell is going to become like this cell, because it's a cycle, right? So the DNA is going to uncoil. These chromosomes are going to become invisible because they're going to start unwinding, because they had to wind up really tightly to become like this. When they unwind, they form a really thin, long string that you can't see. And this is important, right? Because now the cell can use the DNA to make proteins and all these kinds of stuff. Because when it's supercoiled or condensed, the cell can't use it. And again, the nuclear envelope forms, but it kind of started forming here in telophase already. So that's all. That's all for mitosis and cytokinesis. Now there's just some small other details that I need to fill in along the way if you really want to make sure you know everything. Okay, so cytokinesis. Remember, that's this part where the cell divides into two finally. Now, we talked about a guy, right? So we're using a human, an animal. For an animal, the cytokinesis happens kind of like tying a string around a long balloon and making it tighter and tighter and tighter until it breaks into two. Okay, so an animal cell divides from the outside inwards. Okay, a string tightening from the outside inwards. But say we were talking about a plant. For a plant, it would be different. It divides from the inside outwards. So basically, a barrier or a cell plate will form from the inside of this plant cell. And basically, once it formed all the way through, the cell will divide into two. So it divides differently. For a plant cell, it divides from the inside out, whereas from an animal, it divides from the outside in. You don't need to know that many details, just know that they contrast in that way. Okay, here's some words. So we talked about prophase, metaphase, anaphase, and telophase. Here's some words to describe the process. I don't want to include the words on the diagram because it's going to make everything look 20 times more complicated than it should be. So now next part, cyclins. So cyclins is little molecules, okay? Molecules, I call these cyclins, okay? And what they do is they regulate this entire process, right? So coming back to here, this whole process happens over and over again. But why does it happen in this cycle? Why does it happen so smoothly and efficiently? It happens so smoothly and efficiently because of cyclins, okay? What do cyclins do? Cyclins bind to enzymes called cyclin-dependent kinases, okay? So they go like this. They bind nice and smooth. And when they bind, they activate this enzyme, this kinase. And this kinase will then go to its target site and activate this target site. And this target site is what? Guess what? This target site can be a place that causes the G1 phase to be activated, or a place that causes the S phase to be activated, or a place that causes G2 phase to be activated, or a place that causes mitosis to be activated. So that's why these cyclins are so important. These cyclins activate these enzymes called cyclin-dependent kinases, and once they're activated, they will cause a specific outcome by regulating one of the stages of the cell cycle, okay? So that's why cyclins are so important. You don't need to know that much, you just need to know exactly what I just told you. Here's a little graph that basically says what I just said. These cyclins, they will bind to the cyclin-dependent kinases, and they will cause, for example, there's a cyclin called G1 cyclin, and it will cause G1 phase to be activated, you can see. Whereas this G1S cyclin is going to cause the cell cycle to go from G1 to the S phase. Then there's another one called an S phase one, which is going to cause the S phase to be activated, okay? So that's basically the concept. These cyclins are like little machinery, little tools that help activate and make the cell cycle happen in the right order and all this stuff. If these cyclins don't work properly, the cells will become a complete mess and they won't grow properly at all, okay? And this can lead to cancer, which we'll talk about shortly. Speaking of cancer, now is shortly. So here we have cancer, okay? So cancer, this is the last and final thing, by the way. So if you stuck this long, you're definitely going to do well on your test. So here we have a guy, okay? We're looking at his skin, okay? His skin is made up of cells, okay? Cells. Now, what is cancer? So ordinarily, cells have a life cycle, right? Like I said, right? They grow and eventually they die. The process of dying naturally for a cell is called apoptosis. Just like how a human gets old, they die, right? Naturally. Like, if you get too old, you're going to die. Same for a cell. This process is called apoptosis. Now, cancer is a problem where these cells don't die when they should. They keep going and keep going forever. They live very long. Okay, so these cells here, some of them will have cancer, and they will grow and become weird shapes, and they will grow and grow and grow and form a primary tumor. Okay, a primary tumor is a tumor that originates from one place. And in the beginning, it's going to be benign. Benign means it doesn't spread, okay? It's going to stay on the skin. But if it takes too long, I mean, if too much time passes before you can treat it, then this tumor will pass out of the skin. For example, this is a barrier separating the skin from your blood vessels. And so now this barrier will get broken, okay? And this tumor, again, this is a tumor, will start spreading into blood vessels. So this is a blood vessel. And the process of this tumor cell spreading is called metastasis, okay, metastasis. And so it's going to travel down this bloodstream, and eventually it's going to reach other places. And other places include your brain or your bone or whatever. And then when it gets there, it will continue growing there and get bigger and bigger and form an even bigger tumor. And it's called a secondary tumor because this tumor is growing but originated in another place. It originated from another place. It didn't start in the brain. It originated from somewhere else. And we call this a malignant tumor because it's not stuck in one place. It's moving around to different places. Now, how can this happen? How can cells become like this? Many things, okay? One of which is cyclins. If your cyclins aren't controlled well, remember, cyclins are controlled by your DNA. So if your DNA is damaged due to too much sun exposure or things like that, you'll get skin cancer. Or if you ingest dangerous things, many things, even just genetics, maybe it runs in your family, then you may have problems with your cyclins. And so now this process is not controlled properly, and so they grow like crazy, okay? So that's just one of the things. There are many reasons. And that's why cancer is so bad. Now it will grow in your brain and compress your brain. And we know your brain is responsible for making you survive. And if you destroy your brain like that, you will die, okay? But again, there are many types of cancer. And I'll make another video where I talk about this a little bit more detailed. I just want you to understand the gist and understand that although mitosis is very good for us, it helps us, like I said, grow up and heal wounds. If mitosis goes out of control, if cell division goes out of control, it can lead to cancer.